Hello everyone and welcome to the biology lecture series, and today we'll be discussing the topic sexual reproduction in the flowering plants. So this lecture is the continuation of the previous lecture. So in the previous lecture, what did we study? So in the previous lecture, I have introduced you to the topic flowering plants. So flowering plants are basically the angiosperms, okay, and we studied about the importance of the flowers, okay. So flowers, they have a lot of aesthetic value, cultural value, all right, and they are used as the ornamental plants also, such as roses. Now we also talked about the different parts of a typical flower. So there were four parts, there were four whorls, okay, calyx, then petals, then androecium, and then gynoecium. So we talked about the androecium in the previous session, okay, and then within the androecium we talked about the stamens. So stamens are made up of the filament as well as the anther, and then we talked about and that anther is bilobed, and each lobe consists of the two pollen sacs, okay, and the pollen sacs consist of the pollens. Okay, so now in this lecture, what are we going to study? So we'll be studying about the gynoecium. So we'll study about the pistil. So I've told you the pistil, they represents the gynoecium, and gynoecium is the female reproductive part. Now apart from this, we'll also study about the megasporangium. So in the previous lecture, we studied about the microsporangium. So microsporangium, they give rise to the pollen sacs. And in this session, we'll study about the megasporangium. Megasporangium is also known as the ovule. And then we'll also study about the embryo sac. Now let's start this lecture. So gynoecium, it represents the female reproductive part. And the gynoecium is consist of a single pistil, or you can say single carpel. So whenever a single pistil is present, then we call this condition as monocarpellary. So mono means one. So there is only one carpel present. Clear? This is known as monocarpellary. But if the flower or if the gynoecium, it has more than one pistil. Okay, if it has more than one pistil, this condition is known as multicarpellary condition. Means many carpels are present. Okay, next. Now when there are more than one, the pistils may be fused together. So there were two conditions. Either the only one pistil is present, that is monocarpellary, or more than one pistil is present. This is known as multicarpellary condition. In multicarpellary condition, what happens? Again, two conditions can arise. So all these pistils can be fused together. They can be fused together. This is known as syncarpus. So syn means fusion, and then other condition is the apocarpus. Apocarpus means all these pistils are not fused, they are free. Okay, so in this figure you can see here, figure number this B. Okay, figure 7.1b, this is syncarpus condition. Here the pistils are fused together. Okay, this is the syncarpus ovary. Syncarpus ovary means here the pistils are fused together. Syns means fusion, and this above part is known as the stigma. So we have seen the structure of the gynoecium. Okay, gynoecium looks like this. Upper part is known as the receptive part, which is the stigma. Then we have the style, and the swollen part, this is known as the ovary. Okay. Clear? So this is a very rough sketch that I have drawn. This is the ovary. This is the style, and this is the stigma. Okay, so this is the pistil. So if many pistils are present, then it's multicarpellary, and these can be fused or they can be free. If they are fused, then they are known as syncarpus condition. If they are free, then it is known as the apocarpus condition. Here 1.7c image, this shows you the apocarpus condition where these pistils are free. Okay, now let's talk about the parts of the pistil. So pistil has three parts: ovary, style, as well as the stigma. So the swollen base, this is known as the ovary. Okay, and this entire structure, gynoecium, it is attached at the thalamus. Okay, what is thalamus? Thalamus is the swollen part of the pedicel. And what is pedicel? Pedicel is known as the stalk of the flower. Okay, thalamus is the swollen part. You must have seen swollen base. Okay, so it is present at the base and all the floral whorls, okay, they are attached to this thalamus. Clear? It is the swollen part of the pedicel. Then there is a tube-like structure which is known as the style, and then the uppermost part, this is known as the stigma. This is the receptive region, means it receives the pollen. What does this mean? This means this receives the pollen. Here, now next. The stigma serves as a landing platform for the pollen grains, means stigma is the receptive part. Stigma receives the pollen grains, and then at the stigma, the pollen incompatibility or compatibility is decided. Okay, if the pollen is incompatible, then it will not germinate. Clear? Now the style is the elongated slender part beneath the stigma. So you know about the style. Style is a tube-like structure. It is the elongated slender part through which the pollen tube will travel. Okay, through which. So whenever the pollen lands on the stigma, then what happens? If the pollen is compatible, then it will start to germinate. And then when it will germinate, then it will produce a pollen tube. So if this is the, you can say, pollen that is released in the two-cell stage. One is the vegetative, another one is the generative phase. So two-cell stage consists of generative and the vegetative cell. Then this will germinate and it will produce the pollen tube. So this is the pollen tube. And this pollen tube will contain two male gametes. Okay. What is this? This is the pollen. This is what? This is the pollen tube. Then this is the what? This is the nucleus. Male gametes. These are the male gametes. Two male gametes are produced. Clear? Now the basal bulged part of the pistil is known as the ovary. Okay, the basal part is known as the ovary. Now inside the ovary is the ovarian cavity, which is known as the locule. So within the ovary, what is present? Ovule. Ovule is present within the ovary. And inside the ovary, ovarian cavity known as the locule is present. Clear? Or you can also say ovules are present within the ovary. Now the placenta is also located inside the ovarian cavity. Now next, now arising from the placenta are the megasporangia, which is commonly called as ovules. So megasporangia is known as the ovule. So if there is a question, megasporangia represent what? What is megasporangia? Megasporangia, you can say it is a ovule. It is also commonly called as ovule. So megasporangia, it arises from the placenta. So arising from the placenta are the ovules. Clear? Now the number of ovules in the ovary may be 1. Okay, for example in wheat, paddy and mango, or it can be many. Many ovules can be present within the ovary. For example in papaya, watermelon and orchids. And you know, ovules, they give rise to the seeds. And ovary, it gives rise to the fruits. Clear? And if we talk about the angiosperms, the ovules, or you can say seeds, are enclosed within the ovary wall. Clear? Now, so this is an image showing you the ovule. Now the megasporangium, which is also known as the ovule, okay, So this is an image showing you the typical ovule of the angiosperm. So there are two ends, one is the chalazar end, another one is the micropylar end or the micropylar pole. Then you have so the ovules have two integuments, outer integument as well as the inner integument, and these integuments develop into the seed coat. These later on develop into the seed coat. And then you have the nucellus here. Nucellus, the cells of the nucellus are diploid. So when we will study about the embryo sac, we'll talk about the nucellus also. And embedded in the nucellus is the embryo sac. So embryo sac is embedded within the nucellus. Okay. Clear? Here is the, you can say, funicle, okay, which is known as the stalk of the, this is known as the stalk of the ovule. Okay, and the ovule is attached to the placenta through the funicle. This also you have to remember. Clear? Now let's move on. Now, ovule is a small structure attached to the placenta by the means of a stalk known as the funicle. Clear? Funicle I have already showed you in the previous image. It is the stalk of the ovule, and the ovule is attached to the placenta with the help of this funicle. Ovule will give rise to seeds, so you can say seeds, these are attached to the placenta. Now the body of the ovule fuses with the funicle in the region called as the hilum. So you must have seen this hilum. So the body of the ovule means, okay, fuses with the funicle in the region known as the hilum. Clear? Now thus hilum represents the junction between the ovule and the funicle. Now each ovule has one or two protective envelopes known as the integument. So there were two integuments, outer integument and inner integuments, and the integuments encircle the nucellus except at the tip where small openings called the micropyle is organized. Alright, so you must have seen in that image the integuments were not completely surrounding the ovule. So integument encircle the nucellus, which cells in the nucellus were diploid, but except the tip where a small opening called micropyle is present. So integuments, they encircle the nucellus, but the integuments are absent at the tip of the ovule, and this tip where the integuments is absent, this is known as the micropyle. Now opposite the micropylar end is the chalazal end. So one is the micropylar end, another one is the chalazal end, which represents the basal part of the ovule. So micropylar end represents the tip of the ovule and chalazal end represents the basal part of the ovule. Now enclosed within the integument is the mass of the cells known as the nucellus. Okay, so nucellus is enclosed by the integuments. Now cells of the nucellus have abundant reserve food materials. Clear? Now embryo sac, also known as the female gametophyte, this is present within the nucellus. This you have to remember. First of all, you have to remember female gametophyte, this is known as the embryo sac. Okay, male gametophyte, this is known as the pollen grain. Megasporagium is known as the ovule, and microsporagium is known as the pollen sac. So we have megasporagium, then we have microsporagium, then female gametophyte, okay, and then male gametophyte. So megasporagium, this is the ovule. And then I have the microsporagium. This was what? These were the pollen sac. female gametophyte, this is the embryo sac. And the male gametophyte, this is the pollen. Pollen grains are known as the male gametophyte. Clear? Now, an ovule generally has a single embryo sac formed from a megaspore. We'll study about the megasporogenesis in the upcoming slide. So, you have to remember that embryo sac is present within the nucellus and nucellus is present within the ovule. Okay, and an ovule generally consists of a single embryo sac formed from a megaspore. Okay, now let's talk about the megasporogenesis. Now, the process of formation of megaspore from the megaspore mother cell, this is known as megasporogenesis. Now, ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. So, what happens in the micropylar region of the nucellus means the tip of the nucellus, the megaspore mother cells is formed. So, megaspore mother cell is diploid. Okay, because they are formed from the nucellus. Nucellus is also diploid. Now, it is large cells containing dense cytoplasm and prominent nucleus. So, these cells contain dense cytoplasm and prominent nucleus, and megaspore mother cells, it undergo first meiotic divisions. Okay, now first step in the megasporogenesis is the formation of the megaspore mother cell. Then megaspore mother cell undergo the meiotic divisions, means here there will be the reduction of the chromosome number. Okay, so what is the importance of megaspore mother cell undergoing meiosis? So meiosis in the megaspore mother cells results in the production of 4 megaspores which are haploid. Okay, so these are haploid structures, and we have already discussed that this megaspore mother cell is formed at the micropylar end, and it is formed from the nucellus. So you can see this is the nucellus, this is, and megaspore mother cells has been formed. So it has dense cytoplasm as well as the nucleus is also present. So this is the megaspore mother cell which is formed at the micropylar end. Then what happens? There is meiosis in this megaspore mother cells. Okay, so because of meiosis, first 2 cells will form, and in the end 4 cells will be formed. So we know during meiosis, what happens? Meiosis takes place in 2 phases, meiosis 1 and meiosis 2. And in the end, what is formed? 4 cells are formed. 4 unique haploid cells are formed. Okay, so 4 cells, tetrad means 4 cells. So a tetrad is formed after the meiosis. So here meiosis is happening. Meiosis 1 you can say, and then which leads to the formation of dyad, and then meiosis 2. Okay, and after meiosis you get 4 unique haploid cells. Now the female gametophyte, let's talk about the female gametophyte. So embryo sac is known as the female gametophyte. Now in the flowering plants, one of the megaspore is functional. So 4 megaspores were produced. Out of the 4 megaspore, only 1 megaspore is functional, while the other 3 degenerate. Okay, now only the functional megaspore, it further divides and leads to the formation of the female gametophyte, which is also known as the embryo sac. Now this method of embryo sac formation from a single megaspore, this is known as the monosporic development. Okay, so what is monosporic development? Formation of the embryo sac from a single megaspore. So 4 megaspores were produced after the meiosis in the megaspore mother cells. A megaspore tetrad was produced. So all these megaspores should be haploid, and out of these 4, 3 megaspore degenerate and only 1 megaspore, okay, divides to form the entire embryo sac. So what will be the ploidy level of the cells in the embryo sac? It will be haploid. Okay, so all the cells in the embryo sac, these will be haploid. Okay, all the cells in the embryo sac, these will be haploid, means it contains 1 set of chromosome. Clear? Now what will be the ploidy of the cells of the nucellus? Nucellus cells, these are diploid. Megaspore mother cell, these are also diploid. Okay, Functional megaspore will be haploid. Female gametophyte, again the cells will be haploid. Now let us study the formation of the embryo sac in detail. Now this is the figure. Okay, this figure is showing you the nucleus of the functional megaspore divides mitotically to form two nuclei which moves to the opposite poles forming two nucleate embryo sac. This is an image showing you the formation of the embryo sac. So what is this showing? This is showing you the formation of the embryo sac. So embryo sac is formed by the divisions in the functional megaspore. So what is happening? The nucleus of the functional megaspore divides mitotically. Now mitotic divisions will take place because megaspore is haploid, and here because of the mitotic divisions, okay, divides mitotically to form two nuclei which move to the opposite poles forming the two nucleate embryo sac. So initially what happens? A two nucleate embryo sac is formed. Initially a two nucleate embryo sac is formed, and then you can see a four nucleate embryo sac is formed, and then finally 8 nucleate embryo sac is formed, and these 8 nucleus, these are arranged like this. Okay, so this is how the embryo sac looks like. Okay, so this is the chalazal end, means the basal end. The chalazal end consists of the antipodal, and the micropylar end consists of the egg apparatus. So this is the micropylar end. This is the micropylar end. Here you can see one egg cell is present and two synergies are present, and all these cells are haploid, and then in the center you can see there are two polar nuclei present. Okay, so these cells will also be haploid, and then on the chalazal end 3 antipodal cells are present, and all these cells are haploid, and these are produced by mitosis in the functional megaspore. Okay, clear? Now two more sequential mitotic divisions result in the formation of 4 nucleates. This we have seen. So first of all 2 nucleate cells were formed, and then 4 nucleate cells are formed, and later 8 nucleate cells are formed. Okay, now it is of interest to note that these mitotic divisions are strictly free nuclear. Free nuclear, what does free nuclear means? Nuclear divisions is not followed by the cell wall formation. So you can see all the cell, they lie, okay, within the embryo sac. Okay, so embryo sac, it is 8 nucleate. Okay, so after the 8 nucleate stage, cell walls are laid down, leading to the organization of a typical female gametophyte, also known as the embryo sac. So embryo sac, you can say it is 8 nucleate. Okay, and later on what happens? The two polar nuclei, they fuses with each other. The two polar nuclei, they will fuse with each other. Okay, later on during the fertilization, and this will lead to the formation of 8 nucleate embryo sac, which is 7-celled, because the two polar nuclei have fused with each other and they have formed a single cell. So the mature embryo sac is always 8 nucleate and 7-celled. So this is mature embryo sac. Okay, now here you can see this is the mature embryo sac. This is the micropylar end. So this is known as the egg apparatus, which consists of the eggs and the synergid. This is the egg apparatus. Okay, this is the polar nuclei which are haploid. All these cells are haploid, antipodals are also haploid. So this is the organization of the cells within the embryo sac. Okay, and you can see the cell wall is also developed after the free nuclear division. Now you can see 6 of the 8 nuclei, these are surrounded by the cell walls and organized into cells, and the remaining 2 nuclei, known as the polar nuclei, these are situated below the egg apparatus in the large central cell. Now this is the characteristic distribution of the cells within the embryo sac. Now 3 cells are grouped together at the micropylar end, which constitute the egg apparatus. And the egg apparatus in turn consists of 2 synergid and 1 egg cell. The synergids, you should remember that they have special cellular thickenings at the micropylar tip, which is known as the filiform apparatus. And the pollen tube, it enters through this filiform apparatus. Okay, so the pollen tube enter through the micropylar end, and it will first enter the filiform apparatus, which is present in the synergids. Okay, so this play a very important role in guiding the pollen tubes into the synergid. So this is very important, filiform apparatus. Now 3 cells at the chiasm end, these are known as the antibodals, and the large central cell, as mentioned earlier, it consists of 2 polar nuclei, and these 2 polar nuclei later on fuses with each other. So the mature embryo sac, it is 8 nucleate and 7 cell structures. Okay, so this is the end of this lecture. I hope you enjoyed this lecture, and we'll continue with the rest of the topics in the next session. Thank you very much for listening.